Okay, beta, listen please, all of you. There is for the biopracticals, this time in term 2, we are having the experiments, that is a line of experiments that which consist of the experiments which you are to perform. Okay, so this is one of the activities from those A experiments, that is A category, A section of the experiments. B section, that is only to view, that is for spotting, you know, right? So here you are to perform this experiment. One of these experiments is to collect the water from the different water bodies around you and study them for the pH, clarity, and presence of any living organism. We are to collect the water samples. Okay. What we are to do is we collect the water samples here in the school. We can get it one from the pond nearby, right? From some, you can say, locality where the water supply is bad in quality. So you can get that water also. Okay. And also the water which is there in the school water taps, right? So we do take those water, even the water which is stored, and we have a small little pond up on the terrace also of the school, right? So that water samples, they are collected, like 2-3 water samples we collected, and we perform this on the exam day. You will be also performing it in the same manner as you will be seeing it. It's video. For pH, you know, we have to use a pH strip or the universal indicator. Okay. Giving that pH strip, when you dip it into the water solutions, you will be getting certain color change on the pH strip, and you just correlate it with the pH paper, that is all the strip of that paper where you get the color bands. So there you just match it and you give your observation as it is acidic or basic. Okay. Then clarity of the water. That means the water was clear enough or not. At what width or what length do the water provided is clear or not. Basically, this is performed in the water bodies, right? So we are not going to do that for the water bodies. Clarity में आप यहाँ पे कैसे check करते हो? Simple. We just go with the light experiment. लेकिन आपको जो दिखाया जाएगा वो वीडियो में actual method है जिससे clarity जो है वो check करी जाती है. वो अभी मैं आपको दिखाती हूँ थोड़ी देर में. बट यहाँ पे आप लोगों ने performing में क्या करना है? Simply you have to just placing it in front of the light, natural light or the artificial light, and you have to see that whether the water sample was clear enough or it was having some of the, you can say, turbidity inside it, like it was having some whitey, you can say clear, you can say whitish color का थोड़ा सा appearance नजर आता है, ठीक है? यहाँ पे थोड़ा blur नजर आता है, ठीक है? यहाँ it is totally transparent, it is full complete good quality of the water, ठीक है? तो उस हिसाब से आप लोगों ने इसका observation लेना होता है. Then presence of the living organisms. This requires a slide formation. ठीक है, मैं आपको ये सारा साथ में इसलिए बता रही हूँ क्योंकि जो वीडियोस अवेलेबल हैं उनमें कुछ एक चीज डिफरेंट होती है as what we are to perform here in the lab also, right? You are to perform here in the lab. You are not to show video. You are not to follow any videos, right? You are to just perform when your exam will be there. ठीक है? So this presence of any living organism, this requires the slide formation. Slides के लिए हम क्या करते हैं? That is the same method you will be seeing in the video also. हमने सिंपली क्या है, वाटर सैंपल में से कलेक्शन लेनी है वाटर सैंपल की. If the sediments are there, we take those sediments as in concentrated form. If there are no sediments, then simply the water sample as a whole, with the help of the dropper, it is placed over the cover, over the slide, placing a cover slip, viewing it under the microscope. You will be looking onto the different microorganisms which are present. If the water sample has a lot many, you can say microbes which were present in the water sample, what kind of the water sample it is, उसमें जैसे pond water होगा तो बहुत ज़्यादा क्या कहते हैं microbes जो हैं, organisms जो हैं वो आपको उसमें मिलेंगे. तो you will be just writing your observation accordingly that there are number of microorganisms which are available. ठीक है, good amount of the microbes which are available. Whereas in the water that which has been purified for drinking, maybe there are lesser number or comparatively there are no microbes which will be visible in the sample. The very least of the microorganisms will be visible in that sample. So accordingly, you will be making up the table chart. So you have to do this in your practical file also. And in the meanwhile, I start with the video for this experiment. First, watch the video completely, and then we will start with the simulation also. So these are the experiments of your class 12. What we have to do in this term, first is this water sample, second soil sample, and third is mitosis in onion bean. Okay, 3 experiments we have to perform. So this is the one which we have to perform right now. This is in the presence of the water samples. Okay.
presence in water. Water covers about 3/4 of the Earth's surface and it is essential for all known forms of life. Some of the characteristics that control the quality of water are the turbidity of water, pH of the water, and different types of plants, animals, and their density. To study the turbidity of water samples using Xii's disk method, turbidity is the measure of the relative clarity of the liquid. Turbidity of the water body determines the depth to which light can penetrate and thus affects the distribution and photosynthesis of phytoplankton and microphytes. Turbidity can be measured using several methods. The easiest and least expensive method is through a Xii's disk. Materials required: Xii's disk tied with a rope of moderate thickness and a meter tape. Procedure: reach the center of the pond in a small boat. Slowly immerse the Xii's disk into water vertically, holding the rope tightly till the black and white segments of the disk disappear. On reaching a particular depth, the disk becomes completely invisible. Using a pin, mark the length of the rope when the disk appears, say 8 centimeters. Slowly pull up the disk till the black and white segments of the disk just reappear. Using a pin, mark the length of the rope where they just reappear, say B centimeters. Using a meter tape, find the length of A and B. Find the mean length, say X centimeters, of the rope by the following method. X is equal to A plus B by 2. Observation: the value of X represents the depth of the photic zone up to which sunlight penetrates in the water body and photosynthesis takes place. Precaution: students are advised to perform this experiment under the strict supervision of a teacher to prevent danger such as drowning. To study the pH of water.
तो बेटा बिफोर प्रोसीडिंग दिस इज द एक्सपेरिमेंटल क्लैरिटी जो हम लोगों ने पहले किया ना, अभी मैंने बोला था आपको pH क्लैरिटी एंड द प्रेजेंस ऑफ द लिविंग ऑर्गेनिजम्स. सो इसमें से क्लैरिटी पोर्शन जो है दैट वी आर नॉट टू परफॉर्म बिकॉज वी आर नॉट हैविंग सच अ वाटर बॉडी नियर बाय अस to which we can reach and to which we can perform the Xii's disk experiment, right? But at least you should know it actually how it is performed. Okay, the way we are to perform it, we are to say the clarity. Then I have already told you about that, right? So let us see for the pH now.
Water. The pH is a measure of the relative amount of free hydrogen and hydroxyl ions in an aqueous solution. Solutions with a pH less than 7 are said to be acidic, and with a pH greater than 7 are basic. The pH of pure water is 7 at 25 degrees centigrade. The pH value of water sample can be determined by using universal pH indicator solution or pH paper strips. Materials required: water sample from the pond, bore well and backwater, pH paper, universal indicator solution, dropper and a tile. Procedure: let's first check the pH of water sample using pH paper. Take a pH paper strip and dip into the beaker that contains backwater. Keep the wet pH paper strip on the tile. Do the same procedure for borewell water and pond water. Wait for some time to dry the pH paper. Note the color and compare with the color chart given on the broad range indicator paper and get a rough estimate of pH of the sample solution. We can observe that backwater and borewell water has pH 8 and pond water has pH 5. Let's check the pH of the water sample using universal indicator solution. Using a dropper, take some universal indicator solution. Put 5 drops of indicator solution into the beaker that contains the water sample. Note the color developed and compare it with the color chart. We can observe that backwater and borewell water has pH 8 and pond water has pH 5. To analyze living organisms in a water sample, a productive water body has high nutrient concentrations and has a very high density of phytoplankton per unit area. These water bodies have high amounts of nutrients and dissolved oxygen and bear a large number of organisms at different trophic levels. You will observe these kinds of living organisms in the water sample with the help of a microscope. Materials required: a water sample from a pond, test tube, glass slide, cover slip, needle, dropper, filter paper, and a compound microscope. Procedure: take a beaker containing pond water and pour it into a test tube. Leave the water sample undisturbed till the sediment settles to the bottom of the test tube. Decant the pure water, leaving concentrated sediment at the bottom of the test tube. With the help of a dropper, transfer a drop of water from the test tube to the center of a glass slide. Take a cover slip and place it gently on the slide with the aid of a needle. Remove the extra water using a filter paper. Place this glass slide on the stage of the compound microscope and view it. Observations: we can observe a large number of different organisms in water sample.
Anji beta, so this was the video showing you the 3 of these experiments done together. That is to study the, once stop sharing. Anji. So this was the 3 of the experiments done in one, that is to collect the water from the different water samples, okay, from the different water bodies we are having. And I told you that you will be also getting 2 or 3 of these water body samples. And in your exam, testing it for pH, for clarity, and for the living organism. How to perform this experiment, that is okay to you? Any confusions to anyone? Anji bache, any confusion to anyone who wants to discuss anything about this first experiment, that is to collect water from different water bodies around you and study them for pH, clarity, and the presence of any living organism? Here, the presence of living organism, that requires a slide formation, as we have seen in the video also. So this slide, you are to show it also, right, to the examiner when you are to get it checked from the examiner. Then the slide has to be shown on the compound microscope. It has to be shown to the examiners, provided your slide should have that particular organisms on the slide which you are needed to show, and you are needed to give it as an observation that these has a lot many of the microbes and this has a fewer number of the microbes as in comparison to the other sample. Okay, and check is just मैंने आपको पहले बताया, we are not to do that particular experiment here, but you should at least know it how it is done, right, for turbidity check or for the clarity check, right? So this was one of the experiments out of the 3. The another 2 are soil and the mitosis, right? Any confusion, any question anybody wants to ask related to this? Anji bache, anything? Okay, here also we have on the same very slide, we have, let me show you. This is a simulator also, that which shows the number of water samples. You can see here, there are a number of the water samples which has been provided. Checking these water samples with the help of the universal indicator. As we see here is tap water. You can kindly note down these observations side by side onto your notebooks. First for the pH, this is for the pH of the water sample we are doing it, right? Here you write the first sample is tap water. Using the universal indicator, we are providing the 2-3 drops of universal indicator, and that has changed the color into the, what is the color matching to this? I guess 6, right? So that is acidic medium, acidic, right? So it is a bit acidic. You can write observation. आप लोगों ने observation table बनाना है pH के लिए. First write the sample as tap water. plus universal indicator as we have just introduced, and then write its observation, that is, it has attained the pH equivalent to 6, which is acidic. Okay, now let us change this sample. From tap water, we have shifted now to wastewater. This is the second wastewater. Now immersing the same liquid back to this universal indicator. Color has been becoming lightened, but now it is nearly equivalent to what? 4. Anji, kindly tell me if my observation will be a bit different from yours. It's, I guess, equivalent to 4, right? So write this second sample 2, that is wastewater. For the wastewater, the universal indicator added to it results in pH 4, that is acidic. Okay, in the form of a table you will be writing. Next is pond water. Pond water. Again immersing this universal indicator, and it has been changed to 8, a bit basic, right? 7 is neutral. This is 8, right? Now the next is backwater. Again putting the universal indicator, the color has again changed to shifted to 8. pH is same for the previous pond water and backwater, it is 8. Bit basic, right? Little basic. Then for seawater, next sample is seawater. The color remained the same, I guess. It's little change or no change. It's 7, I think. The color is equivalent to 7 or 8, maybe possibly 7 or 8. You can write. It gets somewhere between 7 and 8. Then is pure water. Pure water will be neutral, 7, exactly 7. Sea water was also of the same color. Pure water is also of 7, neutral. Okay. Then the river water, next sample, river water. This is nearly 8. It is 8 pH, and last is the lake water. And this is for lake water again 8. Okay, so this is 8 again. For turbidity, as we know that there was a Seekis disk method. I hope you people have completed it for pH. Now this is Seekis disk. Seekis disk में अगर हम disk का movement ये change करेंगे, you have to see on this right hand corner. Beta, please see on the right hand corner where the length is 0 centimeter. It's written है ना. So as I move the disk inside, as I am increasing the length, you see there is the turbidity that is disappearing of this disk is going on and on and on, right? So this is increasing the length. As I am increasing the length of the rope, the disk has been disappeared. This is nearly at 92. It's visible 94, 93, 94. You know? So this is 94 centimeters totally disappearing, and 92 it was last appeared, है ना? Lightly appeared. तो ये formula लगा के आपने average निकाल लेना है. That is with length of rope as 94 centimeter final length where it disappeared, and 92 as final initial length where it was, you can say, finally appearing. Okay, final stage of its appearing. So 92, right? So that is the turbidity of this water sample, but there are no more water samples given, so we are not doing that, right? Only one water sample is there. Now this is about making of the slide with the sediments, glass and the cover slip. We are to take the sediments from this sediments, but it is not actually filling it. So you can simply say, आप लोगों ने ये देख लिया video में also, that this is dropper. We put it in the sediments. We collect the sediments from the base, okay, from the base of this sample where the sediments have been taking place. These are then kept over the glass slides, covered with the cover slip, and then viewed under the microscope, the compound microscope. Okay, and same way you will be using the wastewater, that is the sedimentations of the wastewater are taken, and then the glass cover slip and putting the slide over the microscope. You will be possibly looking onto the microbes which are present. Okay, so this is how the process or the procedure, the whole procedure for this 3 of the experiments is done, right? It's one experiment performing collectively 3 of the, you can say, other procedures for the same water experiment. Okay, same way you'll be having for soil, that we will be doing in the next practicals, right? Anything you want to ask related with the practical? So you can start up with your practical file work also for term 2. 3 of the experiments for performing and 3 of the 2 or 3, I think 3 of the experiments for spotting. Okay, so next soil samples we'll be doing it next week, the same period on the same day. Any confusion to anyone related to practical?